Well, good afternoon, everyone. I would like to introduce myself. My name is Ivana Ratkaj, and I'm giving this talk instead of our group leader, Sandra Kraljević-Pavlić, who unfortunately could not be here today. First of all, I would like to thank the organizers for inviting us to participate at this symposium. And of course, I would like to thank Mother Nature for calming down a volcano so that the Europe can join you here today. So, yes, I don't know how the picture of Byron Dupuytren got on my slide, because it's quite obvious after the morning session that I will be talking about Klein's disease. So, so my talk will be about microarray expression analysis, so from rats to humans once again. I've been, well, let's say, relatively new to the problem of this disease. I've been dealing with this disease for about 4 years, and one of our theories that we tried to explain the development and the onset of this disease is through the explanation of healing process that goes wrong. Yeah, I don't have financial disclosures. We have no interest. Yeah. None listed anyway, yeah. So we can imagine that this disease is, as I already said, healing processes that went wrong. So in a normal healing process, when you hurt yourself, you have your fibroblasts that sort of migrate to the wounded region thanks to all the cytokines that are being produced, especially TGF-beta, and they differentiate into myofibroblasts, which have, at that stage, a very good characteristics, and that is that they can have a contractive phenotype, so they will close the wound and allow the healing process to commence. After the process is done, they should enter apoptosis and leave a normal tissue behind. Of course, when this doesn't happen, thanks to the proliferative signals and anti-apoptotic capacities, you have pathological changes, and Dupuytren contraction can be developed. So it is a really safe bet to say that this is a multifactorial and polygenetic disease. And I would like to thank all the speakers before me who really nicely characterized and described myofibroblasts' role in this disease and multiple origins. So they do not have to come from the local fibroblasts, but from stem cells and etc. Of course, I will not go into the genetic research done on this disease, from TGF-beta and their transcription factors to the mitochondrial DNA mutations, because I think that we can all agree that there is a strong genetic background. We just don't know what it is yet. Or I think that today, perhaps for the first time, we are truly aware of the complexity of this disease, which is a good start to understanding it. So in our research, we use the primary cell cultures, which are gotten after the surgery and put into a physiological buffer, and when they got to the laboratory, we simply cut them into small pieces and waited for the cells to come out. At first, there's a modest number of cells that is coming out, but after 10 to 14 days, you have enough cells to remove the tissue and leave your cells to proliferate. When they cover the whole cultured platform surface, we isolate total RNA and done a microarray analysis. We use a standard procedure that you can see on this picture, and we use an Affymetrix platform, and we did a whole genome analysis. So when our results, we got approximately 14,500 information from a single chip, and because it's a lot of data to handle, and to be sure that we got the right genes out of those 14,500, we used 2 ways of analyzing those results. First is the MAS5 or MAS5, which is a software that is integrated into our Affymetrix platform, and we compared a diseased chip or sample with a healthy control. And what I didn't mention that from each patient, the diseased tissue was taken out, as well as the control tissue that from the same sample. So we have a matched patient, disease and control. And the second way of analyzing our results, we used a gene spring analysis where we put cell files in it. So we put a raw data, we didn't normalize it at all, and then simply used the algorithms that are available in gene spring analysis. So then we compared those list of genes and found some common genes. The final analysis was done using a gene ontology, where we wanted to see if any of our genes are already known to participate in some important signaling pathways that are introduced into gene ontology. At the end, we got 18 genes. So you can cluster those genes based on their signal intensity, which correlates to their expression. And as you can see, well, on your left is the list of the 18 genes, but you do not have to remember them. What is here important is the fact that you can cluster our genes into 2 clusters, and you can divide first cluster into 2 groups, and you have a second cluster down there. So as you can see, we had only done microarray analysis on 3 patients. So we had 3 diseased and 3 controlled tissue. But to confirm the expression of those 18 genes, we have done an RTPCR analysis and we had broadened our number of patient size to 16 patients. So as a result of RT-PCR analysis, we confirmed the expression of 10 of those genes from the 18 on the list. And here are their names and their functions, but please don't bother yourself with reading their information about the genes. What is important here is colors. So the blue one represents the first group of the first cluster. The purple one our second group of the first cluster, and the yellow one represents the second cluster genes. So here's a better slide when you can see the list of our genes that are confirmed, and you can see their fold change. As you can see, elastine, which is the first one, has the biggest fold change, over 6 fold change, which is it's been very interesting to me because not only that we've done analyzed our RT-PCR by a software REST, but also I've done a manual delta-delta CT comparison method. So you can see that from that there's a big difference between expression of certain gene from patient to the patient. So it happened that in one patient, this gene was overexpressed over a 100-fold change. So that's a pretty high fold change. And as you can see, there's a big difference in expression of certain genes between the patients, which I think this variance is well normal when you're dealing with this complex disease like this, and when you're dealing with biological samples. And we got the tissue from the terminal stage of the disease. So somebody said that it's like taking a snapshot. Well, yes, it is. But I think that you can describe it even better if you imagine a disease like a movie. So we have, I don't know, 1,000 of pictures on a roll. And so your tissue represents the pictures that are on the far end of that film roll, but that picture is not always on the same place. It's somewhere in the frame of the last 20 to 30 pictures, and it's not always from the same place. So that is one of the explanations of why, yes, the gene expression so varies between patients. So we can divide our genes into, let's say, 2 groups. One of them are they interact with actin, with filaments, with extracellular matrix, and good, you expect those genes, that knowing what we know about Dupuytren's disease, and so no harm done there. But our main focus was on the genes that are regulatory and are signaling genes. First of them is THBS1 gene, or thrombospodin 1. Well, as it has been mentioned that TGF-beta is a very important cytokine because it induces fibroblasts differentiation into myofibroblasts, and it is one of the first cytokines being produced when there's some wound and in the body. But so this gene is responsible for interacting with non-active TGF-beta, and it activates him. And it is well established and it is well known that when you activate TGF-beta signaling cascade, you unleash a large number of genes that encode for the proteins that will facilitate survival proliferation, which is, of course, very important for the onset of the disease. The second gene name here is a member of the AMP kinase, which is directly phosphorylated by another very important kinase. It's called AKT, and it also, which has a proliferative effect on the cell, and this gene is directly responsible for stopping apoptotic signals because it can interact with caspase 8 and so stop the progression of apoptosis. The third one, CLV1, well, it is responsible for smooth muscle and non-smooth muscle contraction and regulation. And the last one is growth arrest DNA damage inducible beta. It is, of course, or it is expressed when a lot of different environmental stresses happen and occur. But why is it important to us? Because it has been shown that this gene is responsible for activating another really potent and powerful kinase, P38, which also has proliferative effects on the cell. So one of the key interests of our research is why do myofibroblasts persist in tissue? Why do they not enter the apoptosis? Well, of course, because they have signals to proliferate and they have anti-apoptotic characteristics. So they can then produce alpha-smooth muscle actin. There's a contraction. There's a extracellular matrix protein being produced because of the persistent myofibroblasts. So I think that these data present sort of the possibly important kinases that are responsible for those signals, like P38 and AKT, and that we have sort of, so we have not only have a proliferating signals, but signals that enable myofibroblasts to escape apoptosis. Sort of the two sides of the same coin. So, and, oops, sorry, wrong, yeah. And in the end, in summary, I would like to summarize that yes, it is a multifactorial and polygenetic disease, that we confirmed 10 of those 18 genes that we got as a result of microarray analysis, and that these genes are, that 4 of them at least, are involved in signal transduction pathways that could be responsible for those very important proliferation and anti-apoptotic signals. Our research doesn't stop here. Our goal is to incorporate these proteins encoded by these genes into signaling cascade that could, well, sort of be one of the possible methods and the possible ways of why does this cell progress, what is the onset of this cell, so we can hopefully know at what level do we have to inhibit and stop the signals from reaching the cells and stay in the culture. At the end, I would like to acknowledge well, my mentors and the people who are responsible for introducing me to Dupuytren's paradox of well-established pathology, but very, very elusive etiology. This is the head of our Department of Biotechnology, Professor Krešimir Pavlić, and our group leader, Sandra Kraljević-Pavlić. So thank you for your attention, and if you have any questions, I would be more than happy to answer them. I have 45 seconds left. Yeah.